Welcome to this lecture on the fundamentals of biomolecules. Today, we are going to explore a fascinating and highly dynamic property of the DNA double helix, its ability to denature and renature. If you have ever wondered how our cells read genetic information, or how scientists perform DNA testing in forensic labs, it all comes down to the simple concepts we will cover right now. Let us dive in and see how DNA behaves under changing conditions. Now that we know our topic, let us start with a simple analogy. I want you to think of a common jacket zipper. The two sides of the zipper are just like the two strands of a DNA molecule. The interlocking teeth represent the weak chemical bonds that hold these two strands together. If you pull the zipper down, the two sides separate completely. This is exactly what happens during DNA denaturation when we apply heat. The heat acts like the zipper pull, breaking those weak connections and leaving us with two separate single strands of DNA. It is a simple mechanical analogy, but it perfectly captures the physical reality of what is happening at the molecular level. Building on our zipper analogy, let us look at the formal biochemical process. DNA denaturation is the process where the double-stranded DNA molecule unwinds and separates into two single strands. This usually occurs because of external stress, most commonly high heat or extreme pH levels. This stress breaks the non-covalent hydrogen bonds between the complementary nitrogenous bases. Remember, adenine pairs with thymine and guanine pairs with cytosine. However, it is absolutely crucial to understand that the strong covalent phosphodiester bonds forming the backbone of each strand remain completely intact. Because the backbone is unbroken, the actual genetic code is not destroyed, it is simply exposed. Since the genetic code remains intact, you might wonder if this process is reversible. The answer is yes. If you carefully align the teeth of an open zipper and pull it back up, the two sides lock together perfectly once again. In the world of DNA, this beautiful relinking of the two strands when the temperature cools down is called renaturation, or annealing. It is a highly specific reverse process where the separated single strands find their exact complementary match and reform those hydrogen bonds. This reassociation happens spontaneously, as long as the disruptive stress, like heat, is removed slowly, allowing the original double helix to recreate itself. Now that we understand how DNA unzips and rezips, we must ask, why does this matter so much to us? In nature, this mechanism is essential for life. Our cells must constantly denature short segments of DNA to actually read the genetic code. Think about processes like DNA replication, where a cell copies its DNA before dividing, or transcription, where a gene is read to make a protein. Without the ability to temporarily separate the two strands, the genetic information would remain locked away inside the double helix, completely unreadable by the cell's machinery. Beyond natural cellular processes, this unzipping property is the foundation of modern biotechnology. In modern science and medicine, we exploit this reversible property every single day in laboratories around the world. The most famous application is the polymerase chain reaction, commonly known as PCR. This is the exact technology used in viral testing, forensic DNA fingerprinting at crime scenes, and broad genetic research. PCR relies entirely on artificially heating DNA to denature it and then cooling it to let specific markers renature. By repeating this heating and cooling cycle, scientists can make millions of copies of a single DNA fragment in just a few hours. Knowing that heat separates DNA strands in PCR, we need to know exactly how much heat to apply. When we heat DNA, it does not all separate at once. Instead, it melts over a narrow temperature range. We quantify this transition using a critical value called the melting temperature, denoted as Tm. The melting temperature is defined as the exact temperature where 50% of the DNA strands are separated into single strands and 50% remain double-stranded. This value is incredibly important for scientists programming their thermal cycler machines, as it dictates the exact thermal conditions required for an experiment to succeed. To truly grasp what the melting temperature represents, let us look at this DNA melting curve graph. On the horizontal axis, we have temperature in degrees Celsius, increasing from left to right. On the vertical axis, we measure the relative UV absorbance at 260 nanometers. Single-stranded DNA absorbs more UV light than double-stranded DNA. Notice how the curve starts flat at the bottom left. Here, the DNA is fully double-stranded. As the temperature increases, the curve suddenly shoots upward. This steep rise is the actual melting process, where hydrogen bonds are breaking rapidly. Finally, it levels off at the top right when the DNA is completely single-stranded. That dashed line dropping from the exact midpoint of the steep rise, that is our Tm, the temperature where exactly half the DNA has unzipped. Now that we can visualize the melting temperature, how do we actually calculate it? For short DNA sequences, like the primers used in PCR, that are typically under 20 bases long, we use a simple calculation known as the Wallace rule. The formula states that Tm equals 4 times the sum of G and C plus 2 times the sum of A and T. Let us break down these symbols. Tm is our melting temperature in degrees Celsius. The variables G, C, A, and T represent the exact count of guanine, cytosine, adenine, and thymine bases in the strand. Conceptually, this formula tells us that every guanine-cytosine pair contributes 4 degrees to the melting temperature, while every adenine-thymine pair contributes only 2 degrees. Why? Because GC pairs are held together by 3 strong hydrogen bonds, making them harder to break, whereas AT pairs only have 2. Therefore, if a sequence has more G and C bases, the melting temperature will increase significantly. Before we move on to longer DNA, let us see this formula in action with a concrete example. Imagine a scientist is designing a short DNA primer for a PCR experiment. We are given that the primer is 15 bases long, containing 4 guanines, 5 cytosines, 3 adenines, and 3 thymines. We are asked to calculate the estimated melting temperature. Because this is a short sequence, under 20 bases, the Wallace rule is the perfect tool. First, we substitute our given numbers into the formula, replacing G with 4, C with 5, A with 3, and T with 3. Next, we add the values inside the parentheses, showing we have 9 strong GC pairs and 6 weaker AT pairs. We then multiply the 9 GC pairs by 4 degrees, giving 36, and the 6 AT pairs by 2 degrees, giving 12. Adding these together gives us a final melting temperature of 48 degrees Celsius. Physically, this means the scientist must set their PCR machine to around 48 degrees to allow this specific primer to successfully bind to its target. That example worked perfectly for a short primer, but what if we are dealing with an entire genome? For longer DNA molecules, we cannot simply count millions of individual bases. Instead, scientists use a simplified empirical formula based purely on the overall GC content. The formula is Tm equals 81.5 plus 0.41 times GC. Here, Tm is again the melting temperature in degrees Celsius. The constant 81.5 represents the baseline melting temperature for a hypothetical DNA molecule with 0% GC content under standard laboratory conditions. The variable GC is the percentage of guanine and cytosine bases in the total DNA molecule. This mathematical relationship clearly shows that as the percentage of GC pairs increases, the melting temperature increases linearly by 0.41 degrees for every 1% rise in GC content. If a DNA molecule has a very low GC percentage, the melting temperature will decrease, meaning it will denature much more easily. Now that we have our empirical formula, let us apply it to a real-world biological scenario. A researcher extracts a long genomic DNA sample from a newly discovered bacteria living in extremely hot volcanic springs. We are given that the DNA consists of exactly 60% GC base pairs, and we need to calculate the expected melting temperature. Because we are dealing with a long genomic molecule, we must use the empirical formula. We take our given GC percentage of 60 and substitute it directly into the equation in place of the GC variable. Next, we multiply 60 by the coefficient of 0.41, which gives us 24.6. This tells us the high GC content adds an extra 24.6 degrees to the baseline. Adding this to 81.5 gives us a final melting temperature of 106.1 degrees Celsius. Physically, this extremely high melting temperature makes perfect sense. The bacteria live in boiling hot springs, so their DNA must have a high GC content to remain stable and prevent spontaneous denaturation in their extreme environment. We have covered a lot of ground today, so let us summarize the key takeaways. First, we learned that DNA denaturation is the unzipping of the double helix into single strands by breaking hydrogen bonds, usually through applied heat. Second, we saw that renaturation is the beautiful reversible process where these strands perfectly zip back together upon cooling. Third, we defined the melting temperature, or Tm, as the specific point where exactly half of the DNA is unzipped, and we learned how to calculate it. We discovered that this temperature heavily depends on the number of strong GC base pairs present in the sequence. Finally, remember that understanding this reversible zipper mechanism is not just academic. It is the absolute foundation of modern genetic technologies like PCR, DNA sequencing, and forensic analysis. You now have a solid understanding of how and why DNA melts. Great job!